Hi, I'm Katie Pearson, and I'd like to tell you about a course-based undergraduate research experience developed by the California Phenology Network. The California Phenology Network is a collaboration of 22 California herbaria funded by the National Science Foundation to advance the digitization of herbarium specimens. The California Phenology Network developed a 10-week undergraduate research course to engage students with the newly digitized herbarium specimen data, improve students' biodiversity literacy and scientific engagement, and create novel data on how plants are responding to climate change. Students conduct original research using data they both harvest and create from digitized herbarium specimens that have been aggregated on the California Phenology Network's data portal, CCH2. Specifically, students develop a question about how plants' phenological events, such as flowering or fruiting time, change with climate factors such as temperature or precipitation. Plant phenology, which examines the timing of plants' life history events, such as flowering and fruiting, is a fantastic topic for biology students due to its importance for plants and their communities. Changes in plant phenological events can have cascading effects in an ecosystem, because the timing of flowering and fruiting can influence any organism that relies on flowers or fruits being available at a certain time. For example, pollinators, migratory birds, bears emerging from hibernation, or humans expecting to harvest crops by a certain date can be affected by changes in plant phenological events. Therefore, phenology is of great scientific and practical importance, and thinking about phenological change can promote deeper thinking about the interconnectedness of nature. The overall arc of this phenology research course is fairly simple. Students will gain background knowledge on phenological research using herbarium specimens from core readings, then use that knowledge to define a research question based on the effects of climate on phenological events of a study taxon of their choice. They then score the phenology of the specimens of their study taxon using the new phenological scoring tools we developed in the CCH2 data portal that allow the editor to score for the presence of reproductive structures in a standardized way. The students then georeference some of their specimens to increase their dataset size, and they use provided R scripts to clean and analyze their data. In the end, they present their results as a research poster or paper. We developed all the materials for this course in winter 2020. These include an instructor's guide, syllabus, lecture slides, sample datasets for R demonstrations, assignments, protocols for using the portal and georeferencing, all R code for data cleaning and analyses, and a course assessment for gauging the success of the course. A pilot run of the course was conducted during the spring 2020 quarter at Cal Poly State University. When we were scheduling the course, we had planned to meet in person weekly. However, because of the COVID-19 pandemic, the course was migrated online and shortened to 9 weeks. Still, on the first day of class, all 19 students logged into Zoom, cameras on, eager to discuss the pre-course readings and delve into crafting scientific questions. This being the first implementation of the course, we faced several challenges along the way. We discovered that our current phenological scoring tools in CCH2 require more development so that students can score specimens across many herbaria, and so that students can search for pre-existing phenological data. We also found that training students to georeference, providing guidance on georeferencing, and quality checking the georeferences took a significant amount of time and expertise on the part of the instructor. Finally, the biggest obstacle to the course running as smoothly as possible was having the students use R code. When we initially wrote the code, we put in a great amount of effort to make sure students wouldn't have problems with setting the directory and loading packages and such. Nevertheless, we didn't foresee how R packages might clash with one another, how climate data might be difficult to download all at once, and how individual computers can just act differently with the same code. Despite setbacks due to the global pandemic, the newness of the course, and coding problems, the results of the course were overwhelmingly positive. These are only a few of the final research posters presented at our virtual poster symposium at the end of the class. Students remained engaged every class, and many started to solve code and analysis problems on their own. Even in the midst of seemingly endless troubleshooting, one student remarked, "This class is definitely the most fun and interesting coursework I've ever taken, and I love it." Results from the pre-course and post-course assessments were also exciting. Students reported significant improvements in their understanding of key topics such as reading primary scientific literature, creating a scientific research question, and designing an experiment or analysis to answer a scientific question. Students also reported that they were now more comfortable practicing research skills like manipulating and cleaning data, analyzing data using linear regression, working with herbarium specimen data, and creating a scientific poster. With these great results and guided by feedback we received from the students, Cal Poly and UC Santa Barbara are now working to improve the course to run more smoothly and require less content knowledge from the instructor. First, we are simplifying the research workflow by removing the georeferencing module, which will enable the students to begin conducting analyses earlier in the course, and it will save the instructor significant time and effort. Next, we are developing more materials to help the students understand the phenological and climate variables they will be using in their analyses. We identified that students frequently misinterpreted results of analyses when they didn't fully comprehend the climate variable they were testing. So we hope to improve understanding of these data and the analyses tools up front. We are also developing our phenological scoring tools as previously described, and we are providing more reference materials that students can use to get familiar with manipulating R code, though the goal is to keep students' and instructors' need for coding expertise to a minimum. As such, we're continuing to rework the R code. During the pilot run of this course, we iteratively developed a better version of the code. However, it currently works for only about 80% of the students. And since we want to be able to have instructors who are less familiar with R successfully run the course, we are considering other options such as moving to cloud-based computing to reduce complications. In the future, we hope to develop the course such that it is easily reproducible not only by members of the California Phenology Network, but also other institutions that are interested in incorporating specimen-based research into their biology curriculum. Until then, we're adding the course materials to our project website and to our published resource on cubes. Stay tuned for updates.